Madison is going to talk about the genetic code, revisited genetic code. Dieter?
Can you hear me? Okay. It is really a great pleasure to be back in Madison. I have not been here for a long time, but really all my career I owe to my first visit in Madison 104. So I was postdoc here at Gorin from 62 to 67. And you saw some of these pictures already. Gorin inviting everybody to come in, of course, to work in the Enzyme Institute. And it was really a fantastic environment for a group of enthusiastic postdocs that really learned to conduct research with extreme experimental rigor, but at the same time with a very fair, with great fairness of citation, which in the work that I'm doing now is less common, unfortunately. And these are standards that we all endeavor to really maintain as our careers progress. I also have to say that I saw in Govin's lab, in Govin, how one should look outside, how one should work in a team. And so I have to confess here that while I was Govin's postdoc, I also worked in other people's laboratory. So I worked with Robert Bock and Bernie Weisblum and published some papers. I worked with Jack Strominger and did some work that made me a JBC Classic a few years ago. I worked with Volker Skoog, the plant physiologist that came to the cytokine work. And I also had Bill Dove explain to me how phage lambda works, but as we saw from the talk about Shankar Adya, I certainly didn't understand it. So, in any case, this then was the genetic code published from Koran's laboratory, and you can see Otsuka and Hayatsu are here and our speakers, and it was really great to see the teamwork, and 3 months earlier, in the same journal, Marshall Nürnberg published the genetic code from NIH. The last thing I have to say, we all learned scientific travel. So this was going to the Federation meeting at Truax Field in Madison, and you can see these 4 people here again at this meeting, and it was that really allowed us to be together over the many, many years. And lastly, I would like to say it was Carl Bose who wrote a book on the genetic code in 1967 and was really very much involved in the genetic code since then, who started what I want to tell you about 13 years ago when he called me up and said, Dieter, the genome sequence of methanocoxia naturally has just been published from Crick laboratory. And I, Carl Bose, do not know how cysteine kinase is made. And I said, yes, Carl, I'll do something. And part of my group work, I think I didn't know it would take 13 to 14 years to actually do it. And it really got us in my career into anaerobic biochemistry and anaerobic genetics, something that I never wanted to do. So today's genetic code. In 1968, Crick published the frozen accident hypothesis because he felt when the code evolves, clearly changes have to happen, and this simply means then that proteins will be mutants. And in modern terminology, this would mean that the proteome is unstable. And since he felt this could not be, it was simply the code evolved and now it's an accident and it's frozen, incapable of further evolution. However, already in the 80s and 90s, it was shown there are actually many alterations to the code. So there are 10 codons that changed in the nuclear codes and 16 codons in the mitochondrial code. And Marshall Nirenberg earlier pointed out that the major one was UGA. UGA was actually a tryptophan codon in mammalian and yeast mitochondria, for instance, and which is of course a nonsense codon in a normal code. And in the meantime, we also know 2 additional amino acids. So a lot of speculation and work has gone into the codon reassignment. Since all the codons were assigned already, what is happening? And in this particular table that I don't want to go into, what you see here in blue are positions where amino acid assignations have changed in certain species, not in all of them. And the question that was asked, of course, why did the code really not evolve any further? And given the knowledge that we now have of the very large amount of horizontal gene transfer, one would imagine that the code is a lingua franca, and unless it's the case, you would not receive the benefits from horizontal gene transfer. So the codon reassignment that nature did is stop codons. So the new natural amino acids, selenocysteine and pyrrolysine, are encoded by UGA, selenocysteine, and by UAG, pyrrolysine. And then the question is, looking now, 40 years later, at how can codon meanings change? And the code is, of course, read by transfer RNA, and the tRNA modification is exceedingly important for this, and then by the amino acid synthetases. So, without going into this, the many modified nucleosides that you see, and there are some of them shown here, modified uridines, for instance, in the first position of the anticodon, extend the readability of the code because the tRNA would only read by local hypothesis and by what we know, 2 of the codons. So the amino acid-tRNA synthetases are ancient enzymes, but wait for my last slide. And the amino acid-tRNAs determine incorporation of the canonical and non-standard amino acids. Currently, the expansion of genetic code is a big topical issue, but what is usually not totally realized, that everything depends on this interaction here of the anticodon and the codon. So everything has to go through the amino acid-tRNA, and I don't want to go through details on tryptophan-tRNA synthetase, so this is a cognate pair. But as you know, in 1958, before the knowledge of tRNA and messenger RNA, Crick's adapter hypothesis assumed the presence of 20 canonical synthetases, and this really became the dogma once it was found out that in E. coli there are 20. However, today's knowledge is really that only 16 amino acid-tRNA synthetases conserved in all known genomes. So the question really is, what happened? And what happened is that what is not universally found is a sporogenal tRNA synthetase that's absent in many archaea and bacteria. The terminal tRNA synthetase is absent in all archaea and in most bacteria. The terminal tRNA synthetase is also absent, let's say, in human mitochondria. Cysteine tRNA synthetase is absent in some archaea. That's why Carl always called me. Lysyl-tRNA synthetase is a totally new enzyme in archaea. However, all the cells have a full complement of 20 sets of canonical amino acid tRNA, and so the question is, how is amino acid tRNA made? One is direct amino acylation that the textbook and my last 2 slides said, and the other ones is by tRNA-dependent amino acid transformations, and that's what I will go through very quickly today. So before I come to this, there are 2 additional non-canonical amino acid-tRNA synthetases. And as Marshall Nirenberg told you already, pyrrolysine is a 22nd genetically encoded amino acid discovered by, actually, work of it by Chan and by Crick. And pyrrolysine is a metabolite in some archaea needed for methanogens for methylamine, but it's also found in a few bacteria. And pyrrolysine-tRNA synthetase is specific only for pyrrolysine and then for a specific transfer RNA that recognizes UAG. And this, as I will show you next slide, is structurally did recently on this. This is an unusual, it is an orthogonal amino acid-tRNA synthetase tRNA pair. We now know the crystal structures of all amino acid-tRNA synthetase and tRNAs, and the structure here is different. There's a very intricate specialized synthetase tRNA interaction phase, 30 amino acids are in contact in there, and this is the reason why this particular pair is being used in many laboratories for trying to expand the genetic code, i.e. to change the enzyme to accommodate other amino acids. And this pair cannot be recognized by the normal E. coli setup on mammalian. So the second enzyme, the second amino acid is phosphoserine. So phosphoserine is a necessary precursor for cysteine transformation methanogens. I'll come to this. And phosphoserine synthetase is specific only for phosphoserine and forms set attached to the tRNA cis. So phosphoserine is the phosphate. If I have an OH here, I have serine, or if I have SH here, I have cysteine. While pyrrolysine, this group is added to the lysine moiety. So the tRNA-dependent amino acid transformations actually go back to Marshall Nirenberg in 1968, where Wilcox and Nirenberg showed that there is another way of somehow making gluten-tRNA-gluten. And this was shown in crude extracts in 3 bacilli strains, where glutamic acid is attached, and it's clearly a mismade amino acid, mismade tRNA, and then you come to a correction that makes glutaminol tRNA. This, at the next paper by Wilcox, plus 2 Nature papers we had in the 80s, were really forgotten because it was so bizarre and no one looked at it. However, when the genomes came around, it became clear that this must be the case. And I would like to just take the example of Deinococcus rhabdurans, where there's an asparaginal tRNA synthetase gene that converts, of course, tRNA acid to asparaginal tRNA directly. However, we showed there's also the indirect pathway that tRNA acid, the mismade amino acid tRNA is made, that then is corrected to this. And the summary is that these tRNA-dependent pathways are actually believed, everyone now believes, to evolutionarily precede the amino acid tRNA synthetases. And this is the route to asparaginal tRNA, glutaminol tRNA in most organisms. This is the route to cysteine tRNA in methanogens. And selenocysteine tRNA, of course, has to be made this way because there's no amino acid tRNA synthetase that can distinguish between cysteine and selenocysteine. So selenocysteine has to be made in this fashion. The knowledge that we now have is basically that there is a non-discriminating amino acid tRNA synthetase, glutamyl tRNA synthetase, which recognizes tRNA-gluten and forms glut-tRNA-gluten, which then is transamidated by an enzyme that uses gluten to generate glutamate and ammonia, and ammonia is then amidating with the glutamine. So this is a transamidation in the cell, and this happens to be the essential pathway in most organisms. This is not just, and we showed this genetically in Ethapods, for instance. And if you look at the aminotransferases, there are 2 types, a heterotrimeric type and a heterodimeric type. The heterodimeric type is an archaea. They have a tRNA binding protein, the E subunit, which is a totally isolated protein family. Only the B subunit of the other enzyme is in the same protein family. And the business end is a classical asparaginase, a type 1 asparaginase. And these together make asparaginal tRNA. This one got the makes glutamine, as you'll see. While in the other case, the GAT-CAD enzyme, the business is a normal amidase. And these are found in bacteria, Archaeal chloroplasts, these are in Archaeal. And it simply shows, this was discussed and the code was done by Carl Rose and others, are there any components that are shared between protein synthesis and amino acid biosynthesis, or has there been anything that can be shared? And here clearly there was a recruitment of amino metabolism enzymes to amino acid tRNA synthesis. If you look at the tRNA specificity, and I can't go in details, it's clear that in bacteria, the enzyme can make either glutaminyl tRNA or asparaginal tRNA, or in other cases where it makes both. So in bacteria, there are 4 different scenarios to make asparaginal and glutaminyl tRNA. Let's say in Helicobacter pylori, both activities are there to make both asparaginal tRNA and glutaminyl tRNA 1 E. coli, which got by horizontal transfer glutaminyl tRNA synthetase from eukaryotic cells. This is not necessary. In chloroplasts, glutaminyl tRNA is made this way, and very recently it was shown that in yeast and mammals, in mitochondria, also glutaminyl tRNA is made this way, while in Archaea, it produces glutaminyl tRNA. This is only, I could show you many crystal structures, I have all of them, but I'll show you only this because it comes back to the point that both Marsh and Nielberg made yesterday and that Carl Rose made in his book. This is a co-crystal structure of the GAT-DE enzyme from Methanothermotrophicus, and it's a dimer. But all I want to show you here is that it binds here the tRNA. And it binds the tRNA in a fashion as we do not see it in amino acid tRNA synthetases. The sequence-independent recognition depending on the size of the loop. And what's important is that the anticodon is free. And in most synthetases, the anticodon is recognized by the enzyme. And this was a final argument why the old idea that the codon could somehow stereochemically bind the amino acid is not correct. Because in this particular case, it is of course a glutamine that is made from glutamate while bound here, so the anticodon cannot be part of a stereochemical recognition with an amino acid. And without going into details, just to show you even further how complex it is, in eukaryotic cytoplasm, of course, we have a normal synthetase, and then these were all the eukaryotic chloroplasts have aminotransferase making glutaminyl tRNA, mitochondria make this. In addition, we showed a few years ago, and this is currently a subject of big dispute, that tRNA is imported into mitochondria, and we could show it's imported and it's required for activity, even though human and yeast mitochondrial DNA have all the tRNA genes for minimal codon recognition there. So I leave this here, and we showed this a few years ago, but probably no one knows it. So it actually shows in this particular route that 11 out of 15 lineages of bacteria cannot make the amino acid asparagine, the way the textbook says. Asparagine is only made in this pathway that generates asparaginal tRNA, and then asparagine will be cleaved off. So this is, as I said, one of these are connections between amino acid and protein synthesis via tRNA. Okay. Now we come to selenocysteine. So selenocysteine is the 21st universal amino acid. And as you probably know, selenium is a dietary trace element. Part of it was done in Wisconsin. In proteins, it's found as part of the 21st amino acid selenocysteine. It was identified by Professor Stadtmann in 1976. And it is believed that seleno enzymes are somehow much better, and I don't have time to go into it, than the sulfur homologs, but seleno proteins are essential in mammals. So you have 25 selenium proteins in your body. Some of them have up to 12 seleniums in it, and mouse homolog 17 seleniums in it. And they are all implicated in health and disease, from HIV to cancer to human mood states to everything. So it was interesting to really figure out how is selenocysteine made. And Boeck and Stadtmann, of course, in tremendous work in between 86 and 2000 showed that in bacteria, serine is mischarged by cell tRNA synthetase to selenocysteine tRNA, which then by PLP-dependent enzyme is converted to selenocysteine tRNA, and you can see OH is serine, SEH is selenocysteine. However, unfortunately, the assumption that Boeck and Stadtmann made that this is universal did not turn out to be true. Archaeal and eukaryotes do it differently, and the only question is how do they do it? So this is not working, but we got an idea when we tried to figure out how cysteine is made in Archaeal, in methanogens. And I have to give Enzyme Soll a gratitude, all the credit for this. So he showed, together with proteomics by John Yates, that there are two unique enzymes, Phosphoserine synthetase, a synthetase that I mentioned earlier, that simply put phosphate onto tRNA, cysteine, and then the enzyme that converts this to cis-deaminases, and this then is how it's made. So this gives the idea that selenocysteine is probably made this way, and we simply wanted to find out why do we have this, and it turns out by deletion experiments that we did that methanogens cannot make cysteine unless it goes via tRNA. So this is the second example where amino acid biosynthesis is RNA-dependent and not worked out otherwise. And this then meant that how is it actually made? And then we have to go back to literature. In 1970, work was done, as a matter of fact, also biochemical work, on an enzyme, a kinase, that just finds the serine attaches to selenocysteine to put phosphoserine on here. Phosphoserine kinase. And then the question is there an enzyme that would go further? And when you look, you find a protein that was already discovered 14 years earlier, SLA, soluble liver antigen, pancreas antigen, is a protein that binds to tRNASec, and it was identified as a target antigen of antibodies diagnostic of autoimmune hepatitis. This has nothing to do with selenocysteine biosynthesis, and we are talking to people in Hamburg who are involved in this part of the work. But is this enzyme SEPSEC-S that then converts it? And the answer is yes, we showed this. And both PSDK and SEPSEC-S are essential enzymes. I could show you many crystal structures. This was published recently. That's the first crystal structure of selenocysteine tRNA. I will also tell you what probably not everybody realizes. You do have two types of protein synthesizing systems because selenocysteine tRNA cannot reach the ribosome with EFTU or EF1A. There is a special elongation factor for selenocysteine, which is EFSec in human and cell B. And we now can figure out finally how cell B or how EFSec recognizes this tRNA and why EF1A does not recognize this tRNA. And this is a famous 9-4 structure. There was a huge discussion in the 90s on this, where 9 base pairs of the acceptor stem are stacked onto 4 base pairs of the T-U stem, and this 13 base pair stem is too long, very likely, for EFTU to hold it. Okay. So this is what we now know about how selenocysteine is made, and with this then we know how amino acids attach to tRNA. But before I close, I would like to tell you one more thing. I don't have the slides. I have the slides, but we do not believe, based on what was already long suggested, the amino acid-tRNA synthetase did not evolve with a genetic code. The genetic code and transfer RNA evolved together. We have done studies that would suggest, going together with other studies, that amino acid-tRNA synthetase, the translated code, so to speak, did not evolve at the same time. So the question is, and this is for the RNA Society, what was there before the amino acid-tRNA synthetases that then translated the code? Thank you.
Thanks everybody. So this brings the conclusion of this session. Now we have 15 minutes coffee breaks. In light of this,